Hello, my name is Joshua Mugabi. I welcome you to Applied Genetics. A strain of barley A has a high yield of seeds, but a long stem which is subject to lodging, that is, a flattening of area of the crop. Another strain B has a short, steady stem, but a lower yield. The genotype of variety A is capital H, small s, meaning high yields and long stem. And the genotype of B is small h, capital S, low yield and short stem. A. Show how a plant breeder would cross these varieties to produce a high-yielding, short-stemmed variety. B. Explain why this variety would not breed true. A. Show how the plant breeder would cross these varieties to produce high-yielding, short stem. The plant breeder would pollinate plant A with pollen from plant B or vice versa. The gametes would be for A, HS, and B, small h, capital S, small h, capital S. When the gametes combined in the zygote, the seeds would all have capital H, small h, capital A, small h genotype. Since capital H and capital S are the dominant alleles, the plants will all be high-yielding, short-stemmed variety. That is the F1 variety. Explain why this variety would not breed true. When the capital H, small h, capital A, small h variety produces gametes, these would be capital H, capital S, capital H, smaller h, capital S, or smaller h, which when combined in the zygote would produce 4 varieties of offsprings, some of which might be short-stemmed, high-yielding, short-stemmed, low-yielding, long-stemmed, low-yielding, long-stemmed, low-yielding. Choose from the list of words below to complete the following sentence. In genetic engineering, a dash from one organism is introduced into the dash of any related organism. The words to pick from, we have chromosome, nucleus, gene, protein, genome. Pause the video and fill the words. In genetic engineering, a gene from one organism is introduced into the genome of any related organism. What name is given to an enzyme which is used to cut a DNA molecule at specific sites? An enzyme which is used to cut DNA at specific sites is called a restriction enzyme. What bacterial cell structures are used to carry the genes intended for genetic engineering? The cell structures in the bacteria which carries gene intended for genetic engineering are called plasmids. Name 3 useful products that can be obtained by genetic engineering. Useful products of genetic engineering are chymosin, alpha antitrypsin, hepatitis B vaccine, and insulin. Outline the steps involved in using bacteria to produce human insulin. The gene for insulin is cut from the appropriate strand of DNA using restriction enzyme. Plasmid are extracted from bacteria cells and opened up using the same restriction enzyme, and the insulin gene is inserted in the gap. The recombinant plasmids are inserted into the bacteria, which then produces insulin. Give 3 examples of genetic engineering that are intended to improve crop plants. Genetic engineering of crop plants can improve resistance to pests, retard ripening, improve uptake of ions from the soil, increase the vitamin content of the crop. In the future, genetic engineering might improve drought resistance and salt tolerance. DNA can be split into fragments using restriction enzymes. A. Outline the technique used to separate these fragments. B. What property of DNA fragments allow this separation? DNA fragments in solution are placed at one end of a sheet of gel, and an electric current is applied which separates the fragments. Then what property of DNA fragment allow this separation? The size of fragments determines how rapidly they move through the gel. Smaller fragments travel faster and farther than larger fragments. Rule horizontal lines through the DNA bands from the crime scene to cross the 3 suspect profile. A. Which suspect is most likely to be guilty? B. Explain why you think so. Given that our DNA bands are here, V, this is got the profile of the victim. Then we have the sample that is got from the crime scene. Then we have suspect 1, DNA, suspect 2, and suspect 3. So we are supposed to use the sample got from the crime scene to eliminate 2 of the suspects, and we pick one suspect who was at the scene. Part A, which suspect is most likely to be guilty? Suspect 3. If you look at the crime scene sample and you look at suspect 3 and you cross the bands, you can see that the bands are corresponding with that one of suspect 3. So we eliminate suspect 1 and suspect 2 because most of the bands from the crime scene sample are not correlating with suspect 1 and suspect 2, but they correlate with the suspect 3. If you look at the different lines drawn, you can see they are correlating. So suspect 3 is the most likely to be guilty. Part B, explain your thinking, why you think so. The greatest correspondence in the position of the bands at the crime scene is with those of suspect 3. Not all the lines, all the relevant lines are drawn here. What is the special characteristic of stem cells and embryonic stem cells? Stem cells can continue to divide throughout their life. The special characteristic of embryonic stem cells is that they can develop into any kind of cell, and in some animals, for example amphibians, into a complete organism. What are the possible sources of human embryonic stem cells? Embryonic stem cells can be derived from the individual cells of an early embryo, from the blood cells in the umbilical cord of mammals, and from the cultures of stem cells. What would be the advantage of using a patient's own stem cells, for example, blood stem cells, to treat his or her illness? The advantage of using stem cells from the patient being treated is that they will not be rejected by the patient immune system. Thank you very much for participating in Applied Genetics, a self-assessment program. Please do like, share, and subscribe.